Welcome, everyone. It's wonderful to have you here. Before we get started, would you let us know in the comments where you're tuning in from and the time of day it is for you? I love seeing how this space brings together people from across the globe. A heartfelt thank you as well to all who've subscribed and supported the channel. It helps me know what connects with you, and it helps others discover these calming moments too. And if inspiration strikes, please feel free to leave your ideas for future videos. With that said, let's begin. Chapter 1. Understanding Life's Chemical Foundations. In the vast expanse of our universe, the essence of life begins at its most fundamental level, the building blocks of life, atoms, and molecules. This intricate foundation serves as the starting point for understanding biochemistry, a remarkable field that explores the chemical processes and substances that underpin all living organisms. Biochemistry is not merely a collection of facts. It is the language through which life expresses itself, revealing the secrets of existence and the connections that bind us. Imagine for a moment the atoms that form the basis of every living thing. These tiny entities, although invisible to the naked eye, are the architects of life. Among them, 4 essential elements stand out: carbon, hydrogen, oxygen, and nitrogen. Carbon is the backbone of organic molecules, possessing a unique ability to bond with itself and a variety of other elements, creating diverse structures that are vital for life. As the eminent chemist Linus Pauling expressed, "The nature of the chemical bond is the most important key to understanding the structure of matter." Hydrogen, the simplest of all elements, is abundant in the universe. It readily forms bonds with carbon and oxygen, contributing to the formation of water and organic compounds. Water, the quintessential solvent, is crucial for biochemical reactions. Oxygen, often regarded as the breath of life, is essential for cellular respiration, a process that releases energy from food. Nitrogen, while often overlooked, is a key component of amino acids and nucleotides, the building blocks of proteins and nucleic acids. Together, these elements form a delicate balance, serving as the foundation for the molecules that orchestrate the symphony of life. Bonds between atoms play a pivotal role in determining the structure and function of molecules. Covalent bonds formed when atoms share electrons create stability and complexity in molecular structures. The strength of these bonds allows for the formation of large macromolecules, which are essential for life. Ionic bonds, on the other hand, result from the transfer of electrons, creating charged particles that can attract or repel one another, influencing various biochemical interactions. Imagine the beauty of these interactions as a gentle weave, where each thread is a bond, connecting atoms into larger structures. This weaving process is not random. It is guided by the principles of chemistry, creating molecules that are specifically suited for their roles in the body. Biomolecules, the organic compounds that sustain life, can be broadly categorized into 4 groups: carbohydrates, proteins, lipids, and nucleic acids. Each of these groups holds unique properties and functions that contribute to the intricate machinery of living organisms. Carbohydrates, often referred to as sugars, serve as a primary energy source for cells. They exist in various forms, from simple sugars like glucose to complex polysaccharides like starch and cellulose. Their versatility and structure allows them to fulfill multiple roles, from energy storage to structural components in cells. Proteins, the workhorses of the cell, are composed of amino acids linked by peptide bonds. Their diverse structures, from fibrous to globular, enable them to perform a wide array of functions, including catalyzing biochemical reactions, facilitating transport, and providing structural support. The renowned biochemist Francis Crick once remarked, "Proteins are the machines of the cell." Lipids, a diverse group of molecules, play critical roles in energy storage, insulation, and the formation of cell membranes. Their hydrophobic nature allows them to form bilayers, creating barriers that protect the internal environment of cells. Nucleic acids, such as DNA and RNA, are the blueprints of life. They carry the genetic information necessary for the growth, development, and reproduction of all living organisms. The double helix structure of DNA, as discovered by James Watson and Francis Crick, is a masterful arrangement that encodes the instructions for life itself. As we delve deeper into the world of biochemistry, we begin to unveil the complexity of life's chemical foundations. Each molecule, each bond, and each element plays a vital part in the grand narrative of existence. We are reminded of the interconnectedness of all living things, a profound truth that resonates through the ages. What does it mean to understand life at this chemical level? It means recognizing that beneath the surface, beauty of nature lies a world of intricate relationships, where atoms and molecules engage in a continuous dialogue, shaping the very essence of life. Through the lens of biochemistry, we can appreciate the elegance of life's design, leading us toward a deeper connection with the universe around us. In this exploration, we encounter not just the science, but also the poetry of life itself. The rhythmic pulse of chemical reactions, the silent whispers of molecules interacting, and the vibrant dance of atoms coming together to form the diverse forms of life we see today. Each aspect beckons us to imagine, to ponder, and to marvel at the wonders of our existence. As we embark on this journey of understanding, let us remain curious and open to the myriad possibilities that lie within the realm of biochemistry. The vibrant world of molecules awaits, ready to reveal its secrets to those who seek to listen and learn. As we turn our gaze to the essential elements that form the bedrock of life, we find ourselves drawn to carbon, hydrogen, oxygen, and nitrogen. These four elements are the cornerstone of biochemistry, comprising the building blocks that interweave to create the vast array of organic molecules. Carbon, with its remarkable ability to form 4 covalent bonds, serves as the backbone of organic chemistry. Picture a spider spinning an intricate web, carbon atoms linked together in chains and rings, fostering an endless variety of structures. This versatility allows for the formation of complex molecules, each serving different functions vital to life. Hydrogen, the simplest element, is no less important. It acts as a bridge between carbon and other atoms, enabling the formation of diverse organic compounds. Imagine a gentle breeze carrying whispers of potential. Hydrogen bonds play a crucial role in stabilizing the structures of proteins and nucleic acids, gently holding them in their functional shapes. It is through the interactions of these elements that life flourishes, each bond contributing to the symphony of existence. Oxygen, a life-giving element, is essential for the process of cellular respiration. It serves as the final electron acceptor in the electron transport chain, a series of reactions that occur within the mitochondria of our cells. As the late biochemist Albert Szent-Györgyi once said, "Life is not a static thing. It is a process." Oxygen fuels the transformation of glucose into energy, allowing cells to perform their myriad functions. Can you imagine the dance of electrons as they traverse the membranes, generating the energy that powers our every heartbeat? Nitrogen, the fourth pillar of life, is integral to the formation of amino acids, the building blocks of proteins. The air around us is rich in nitrogen, yet it remains largely inaccessible to most living organisms. It is the remarkable process of nitrogen fixation, carried out by certain bacteria, that converts atmospheric nitrogen into a form that plants can absorb. This transformation is akin to a hidden treasure being unearthed, providing the nutrients essential for growth. As the renowned scientist Sir Frank Whittle noted, "The world is in the throes of a revolution in the ways we think about the universe. In understanding nitrogen's role, we grasp the intricate connections that sustain life." With these fundamental elements in mind, we transition to the fascinating realm of biomolecules: carbohydrates, proteins, lipids, and nucleic acids. Each class of biomolecule plays a distinct role, contributing to the complexity of life. Carbohydrates, for instance, are often referred to as the body's primary source of energy. Imagine the warmth of sunlight filtering through leaves, Plants harness this energy through photosynthesis, converting carbon dioxide and water into glucose. This simple sugar serves as fuel for nearly every organism, a sweet source of sustenance that powers cellular processes. As we explore carbohydrates further, we find that they are not just energy sources, but also vital for cellular recognition and signaling. When you think of the intricate dance of life, can you envision how cells communicate, sending messages through carbohydrate structures on their surfaces? These oligosaccharides act as identifiers, enabling cells to recognize one another and form tissues, much like a community coming together. The work of scientists like Gertie Corey and Carl Corey has illuminated the pathways of carbohydrate metabolism, revealing the profound significance of these seemingly simple molecules. Proteins, the workhorses of the cell, deserve our attention next. They are formed from chains of amino acids, each sequence uniquely determining the protein's structure and function. Picture a masterful artist crafting a sculpture from clay. The arrangement of amino acids gives rise to an elegant three-dimensional form. Proteins serve a multitude of roles, from catalyzing biochemical reactions as enzymes to providing structural support within cells. The journey of protein synthesis is a remarkable tale, where DNA encodes instructions that are translated into functional proteins, each fulfilling a specific purpose within the intricate machinery of life. As we contemplate the importance of proteins, we cannot overlook the contributions of notable figures in the field. The words of Linus Pauling resonate deeply. The best way to have a good idea is to have a lot of ideas. This spirit of exploration and innovation has driven advances in our understanding of protein function, paving the way for therapies that target diseases at the molecular level. Lipids, another vital class of biomolecules, play an essential role in cell membrane structure and function. These molecules, which include fats, phospholipids, and steroids, create barriers that protect and compartmentalize cells. Imagine a serene lake, its surface reflecting the sky. Similarly, lipid bilayers form protective layers that keep the internal environment of a cell distinct from the outside world. The fluidity of these membranes is crucial for cellular communication and transport, allowing nutrients and signals to pass through. Lipids are not just structural components, they also serve as energy reserves and signaling molecules. When we think of energy storage, we envision the rich reserves of fat that organisms accumulate, serving as a source of fuel during times of scarcity. The intricate balance of lipids in our body reflects a dynamic interplay between energy needs and metabolic processes. The late biochemist Georges-Émile Palade once expressed, "In science, there is a great deal of evidence that something is true, but there is never absolute certainty." This acknowledgement of the complexities of life reminds us of the ongoing journey of discovery. Lastly, we arrive at nucleic acids, the blueprints of life. DNA and RNA, though structurally distinct, work harmoniously to encode, transmit, and express genetic information. In every organism, the sequence of nucleotides in DNA carries the instructions for building proteins, the very essence of life. Can you envision the elegant double helix twisting and turning as it unravels to reveal the secrets of heredity? The processes of replication, transcription, and translation create a continuous cycle of life, where genetic information is passed from one generation to the next, shaping the evolution of species. As we ponder the significance of nucleic acids, we may reflect on the words of Francis Crick. Biologists must constantly keep in mind that what they see was not designed but rather evolved. This understanding of evolution underscores the remarkable adaptability of life, a testament to the resilience and ingenuity of organisms over time. In this exploration of life's chemical foundations, we are reminded of the profound interconnectedness of all things. Each element, each molecule plays a part in the grand narrative of existence, weaving a story that transcends time and space. The beauty of biochemistry lies not only in its scientific intricacies, but also in its capacity to inspire wonder and curiosity. As we continue our journey through the wonders of life, let us remain open to the mysteries that await, For in the realm of biochemistry, every question leads to new avenues of discovery. As we reflect on the intricate tapestry of life's chemical foundations, it becomes increasingly clear that every biomolecule, from proteins to nucleic acids, plays a pivotal role in sustaining the delicate balance of existence. With this understanding, we turn our attention to another essential class of biomolecules, carbohydrates. These vital compounds not only serve as energy sources, but also participate in cellular recognition and signaling, further enriching the narrative of life. In the next chapter, we will delve into the diverse structures of carbohydrates, exploring their significance in energy storage and their impact on health. Prepare to discover how these seemingly simple sugars are fundamental to the complex processes that fuel life itself. Chapter 2. Exploring carbohydrates, energy sources for life. Carbohydrates are often viewed as mere sources of energy, yet they are so much more. They are the fundamental building blocks of life, intricately woven into the very fabric of our biology. Imagine for a moment the sweet, delicate structure of a sugar molecule, a single unit known as a monosaccharide. These simple sugars, like glucose and fructose, are the starting point of a vast and intricate hierarchy that leads to more complex forms, such as disaccharides and polysaccharides. As we delve deeper, we discover that monosaccharides are the single units that combine to form disaccharides, like sucrose, which is formed from glucose and fructose, or lactose, the sugar found in milk. When multiple monosaccharides join together, they create polysaccharides, such as starch, glycogen, and cellulose. Each of these structures serves distinct purposes within a living organism. Picture a tree, strong and steadfast, its roots gripping the earth, while its leaves rustle gently in the breeze. In this metaphor, cellulose, a polysaccharide, provides the rigidity that supports the tree's structure. It is the most abundant organic polymer on Earth, forming the cell walls of plants and giving them the strength to reach toward the sky. But carbohydrates are not merely structural, they are vital energy sources. When we consume carbohydrates, our bodies break them down into glucose, a simple sugar that fuels our cells. This process begins in the mouth, where enzymes in saliva start to digest starches, and continues in the stomach and small intestine. Envision glucose as a tiny spark, igniting the engines of our cells, driving the processes necessary for life. The functions of carbohydrates extend far beyond energy supply. They play a crucial role in energy storage as well. In plants, excess glucose is stored as starch, while animals store it as glycogen, primarily in the liver and muscles. This stored energy can be quickly mobilized when our bodies need an instant boost, like during a sprint or a moment of excitement. As we explore the world of carbohydrates, let us not overlook their importance in cellular recognition and signaling. Carbohydrates are often found on the surfaces of cells, forming complex structures known as glycoproteins and glycolipids. These molecules play pivotal roles in cell-cell communication. Picture a bustling city, where each building represents a different cell, and the interactions between them are facilitated by the signs posted on their walls. These signs, composed of carbohydrates, allow cells to recognize each other and respond appropriately. Consider the words of the renowned biochemist George A. Olah, who once said, "The chemistry of life is fundamentally the chemistry of carbon." Though he focused on carbon's role, carbohydrates, being organic compounds composed of carbon, hydrogen, and oxygen, echo this sentiment. They are integral to the very essence of life, bridging the gap between simple building blocks and the intricate processes that sustain living organisms. Dietary sources of carbohydrates abound in our surroundings, providing not only energy but also vital nutrients. Think of the colorful array of fruits, vegetables, grains, and legumes that grace our tables. Each of these foods offers a unique combination of carbohydrates, fiber, vitamins, and minerals. Whole grains, for instance, provide a treasure trove of nutrients, while fruits and vegetables offer natural sugars along with fiber, which aids in digestion and helps maintain a healthy gut. But not all carbohydrates are created equal. The distinction between simple and complex carbohydrates is essential for understanding their effects on health. Simple carbohydrates found in sweets and sugary beverages can provide quick bursts of energy but may lead to crashes if consumed excessively. In contrast, complex carbohydrates, often rich in fiber, release energy more gradually, keeping our blood sugar levels stable and providing sustained energy throughout the day. Imagine a serene meadow where wildflowers bloom and the air is filled with the scent of fresh grass. This idyllic scene can be likened to a balanced diet rich in complex carbohydrates. Just as the flowers flourish in harmony with their environment, our bodies thrive when nourished with wholesome foods that support our energy needs, without the spikes and crashes associated with processed sugars. The impact of carbohydrates on health extends beyond energy levels. Research has shown that diets high in fiber can reduce the risk of chronic diseases, including heart disease and diabetes. Fiber, primarily found in whole grains, fruits, and vegetables, acts like a gentle broom, sweeping through our digestive tract, promoting regularity, and fostering a healthy gut microbiome. The words of nutritionist Marianne Nestle resonate here. You don't have to be a fat scientist to know that if you eat more calories than you burn, you will gain weight. As we take a moment to reflect on the importance of carbohydrates, we can see the intricate connections between our diet, our health, and the very essence of life itself. Carbohydrates are not just energy sources. They are vital players in the symphony of biological processes that sustain us. Imagine your body as a finely tuned machine, where carbohydrates serve as one of the essential fuels powering your every movement and thought. This fuel not only provides energy, but also contributes to cellular communication and overall health. As we continue on this journey of exploration, let us keep in mind the balance that carbohydrates bring to our lives. They are an integral part of a healthy diet, offering nourishment, energy, and the building blocks for cellular functions. In every bite of bread, every spoonful of rice, and every juicy piece of fruit, there lies a world of chemistry, waiting to be discovered and appreciated. Carbohydrates remind us of the delicate interplay of biology and nutrition, the dance of energy and structure that sustains life. They are more than just molecules. They are essential components of our existence, woven into the story of every living being. As we embrace the wonder of carbohydrates, we find ourselves on the path toward understanding the very essence of life itself. Within the realm of carbohydrates, we encounter a fascinating array of structures, each finely crafted to fulfill specific roles. The simplest forms are monosaccharides, single sugar molecules like glucose and fructose. These organic compounds, with their 6 carbon atoms and an intricate arrangement of hydrogen and oxygen, are the fundamental building blocks of life. Picture them as the single notes of a grand symphony, harmonizing to create the complex melodies of more intricate carbohydrates. As we delve deeper, we find disaccharides, which emerge when 2 monosaccharides join together through a process called dehydration synthesis. This reaction removes a molecule of water, allowing the 2 sugars to bond. Sucrose, the sugar we commonly use in our kitchens, is a prime example, formed from glucose and fructose. It's as if 2 musicians, each holding their own melody, come together to create a beautiful duet. These simple sugars not only satisfy our sweet cravings, but also serve as quick energy sources for the body. Then, we encounter oligosaccharides, which consist of a small number of monosaccharide units linked together. Found in legumes, onions, and garlic, these complex sugars play a critical role in supporting our digestive health. They act as prebiotics, nourishing the beneficial bacteria in our gut. Imagine these oligosaccharides as the gentle caregivers of our microbiome, fostering a thriving community that aids digestion and boosts our immune system. As we ascend the ladder of complexity, we arrive at polysaccharides, the grand structures built from numerous monosaccharide chains. Starch and glycogen are two notable examples. Starch, a storage form of energy found in plants, is composed of long chains of glucose molecules. When we consume starchy foods, our bodies break them down into glucose, providing a steady supply of energy. It's akin to a reservoir of fuel, waiting to be tapped into as needed. On the other hand, glycogen serves as the energy reserve for animals, stored primarily in the liver and muscles. When energy demands spike, such as during vigorous exercise, glycogen is swiftly converted back into glucose, ready to power our movements. As the renowned biochemist Richard J. Roberts once said, "Biochemistry is the study of the chemistry of life." And indeed, these carbohydrate structures embody the very essence of energy management in living organisms. Beyond merely providing energy, carbohydrates also play a pivotal role in cellular recognition and signaling. Glycoproteins and glycolipids, which are carbohydrates attached to proteins and lipids, respectively, are crucial for cell communication. Imagine a bustling city where each building bears a unique address. These glycoproteins serve as the addresses on the surface of cells, guiding interactions and responses. They enable cells to recognize each other and facilitate essential processes such as immune responses and tissue formation. The intricate nature of carbohydrates allows them to convey information, much like a language spoken between cells. This ability to communicate is vital, as it influences everything from immune defense to hormonal signaling. The insights of biochemist Gerald M. Edelman resonate here. The brain is a kind of social organ. It is a network of networks. In this sense, carbohydrates act as the connective tissue, weaving together the myriad interactions that sustain life. As we ponder the dietary sources of carbohydrates, we find ourselves surrounded by an abundance of options. From the wholesome grains that form the basis of many cultures to the vibrant fruits that grace our tables, carbohydrates are ever present. Each source offers a unique composition of sugars, fibers, and nutrients, contributing to our overall health. Whole grains, for instance, are not just rich in carbohydrates, but also a treasure trove of vitamins, minerals, and antioxidants. They symbolize the bounty of nature, nourishing our bodies while inviting us to savor each bite. But how do these choices impact our health? The answer lies in the quality of carbohydrates consumed. Refined carbohydrates, often found in sugary snacks and white bread, can lead to rapid spikes in blood sugar levels, followed by crashes that leave us feeling drained. In contrast, complex carbohydrates, abundant in whole foods, provide a gradual release of energy, promoting sustained vitality. It's essential to consider the source of energy we fuel ourselves with, as it shapes our well-being. To visualize the impact of our carbohydrate choices, imagine a garden. Whole grains, fruits, and vegetables are the vibrant flowers, flourishing under the sunlight of a balanced diet. Refined carbohydrates, however, can be likened to weeds, choking the life out of the garden if left unchecked. Choosing nutrient-dense carbohydrates not only fosters physical health, but supports mental clarity and emotional balance. In this exploration of carbohydrates, we uncover a world of scientific wonder, a testament to the elegance of life's design. As we embrace the role of carbohydrates in energy supply, cellular recognition, and overall health, we begin to understand the remarkable interconnectedness of our biological processes. Each carbohydrate molecule tells a story, a narrative that intertwines with our own, reminding us of the delicate balance we must maintain in our diets and our lives. So, as you reflect on your next meal, consider the journey of carbohydrates that brought it to your plate. Appreciate the energy they provide, the roles they play in communication, and the harmony they contribute to your body's intricate workings. The beauty of carbohydrates lies not only in their chemical structure, but in their profound impact on the essence of life itself. What stories will the next bite reveal to you? As we savor the stories woven into each carbohydrate, it becomes clear that our journey toward optimal health is a tapestry of various nutrients, each playing a vital role. Just as carbohydrates provide the energy and foundation for our bodies, proteins emerge as the essential workhorses, driving countless processes within our cells. In the next chapter, we will delve into the intricate world of proteins, exploring how amino acids come together to form complex structures and the remarkable functions they serve in metabolism and beyond. Prepare to uncover the fascinating mechanisms behind protein synthesis and the vital roles these macromolecules play in the grand narrative of life. Chapter 3: Proteins, the workhorses of cells. Proteins are the essential building blocks of life, often referred to as the workhorses of the cell. They perform a myriad of functions that are crucial for the survival of all living organisms. Imagine for a moment a bustling city where various workers carry out their tasks to keep everything running smoothly. Each worker has a specific role, just as proteins do within our cells. They carry messages, build structures, and catalyze reactions. At the heart of proteins are amino acids, the fundamental units that come together in intricate sequences to form a diverse array of proteins. There are 20 different amino acids, each with unique properties, contributing to the vast complexity of protein structures. The journey of a protein begins with its primary structure, a simple linear chain of amino acids linked by peptide bonds. This initial sequence dictates how the protein will fold and ultimately function. As we delve deeper, envision the transformation from primary to secondary structure, where the chain begins to twist and turn. The hydrogen bonds between the amino acids cause it to form alpha helices and beta sheets. This is a crucial stage as the shape of the protein starts to emerge, much like an artist shaping a clay sculpture. Next, the protein achieves its tertiary structure, where it folds into a three-dimensional shape, stabilized by interactions among the side chains of the amino acids. This folding is not random. It is guided by the chemical properties of the amino acids, resulting in a unique configuration that is vital for its function. In some cases, proteins come together to form a quaternary structure, where multiple polypeptide chains interact to create a complex functional unit. Consider hemoglobin, the protein responsible for transporting oxygen in our blood. Its quaternary structure allows it to carry 4 oxygen molecules at a time, demonstrating the elegance of protein design and function. Once synthesized, proteins do not remain static. They undergo a delicate process of folding, aided by molecular chaperones. These helpers ensure that the proteins achieve their correct shapes, preventing misfolding that can lead to diseases. The intricate dance of protein synthesis and folding is essential for maintaining cellular health. Proteins also serve as enzymes, biological catalysts that accelerate chemical reactions. Enzymes are a prime example of how proteins can influence metabolic pathways. Imagine a bustling factory, where each enzyme is a machine, speeding up the production line. Enzymes lower the activation energy needed for reactions to occur, allowing processes such as digestion and energy production to happen efficiently. One could ask, why are enzymes so important in metabolism? The answer lies in the vast array of biochemical reactions occurring within living organisms. From breaking down food to generating energy, enzymes facilitate these processes with remarkable specificity and efficiency. Consider the enzyme amylase found in our saliva. It initiates the digestion of carbohydrates right in our mouths. As we chew, amylase breaks down starches into simpler sugars, paving the way for further digestion in the stomach and intestines. This is just one example of how enzymes play pivotal roles in our daily lives, often without us even realizing it. The diversity of proteins extends beyond the realm of enzymes. Structural proteins, such as collagen, provide support and shape to tissues in our bodies. Collagen, a fibrous protein, is found in our skin, bones, and connective tissues, contributing to their strength and elasticity. As we age, the production of collagen decreases, leading to visible signs of aging. Another remarkable example is keratin, a protein that forms the structure of hair, nails, and the outer layer of skin. Imagine the resilience and protective functions of keratin. Much like the armor of a knight, safeguarding against environmental damage. Proteins also have roles in signaling pathways, facilitating communication between cells. Hormones, which are proteins, travel through the bloodstream, sending messages that regulate various physiological processes. Insulin, for instance, is a hormone that helps control blood sugar levels. When we consume food, insulin is released from the pancreas, signaling cells to absorb glucose. This elegant system of communication illustrates the vital role proteins play in maintaining homeostasis within our bodies. In the world outside our bodies, proteins are equally fascinating. Consider the enzyme rubisco, which plays a crucial role in the process of photosynthesis in plants. It catalyzes the reaction that incorporates carbon dioxide from the atmosphere into organic molecules, allowing plants to create the energy they need to grow. This highlights the interconnectedness of life on Earth, as the proteins in plants ultimately sustain the animals that rely on them. As we reflect on the myriad roles of proteins, one cannot help but be in awe of their complexity and versatility. They are not merely static entities. Instead, they are dynamic participants in the grand orchestra of life. Each protein, with its unique sequence of amino acids, contributes to the symphony of cellular functions, ensuring that life continues to thrive. Imagine a world without proteins. It would be a silent place, devoid of movement, growth, and communication. Through their remarkable abilities, proteins uphold the very essence of life, reminding us of the intricate beauty found within the microscopic world of our cells. As we delve deeper into the world of proteins, it becomes essential to understand their structure. Proteins are made up of amino acids, the building blocks that come together in chains, folding and twisting into intricate shapes. Each protein's unique function is determined by its specific sequence of amino acids, which can vary dramatically among different proteins. The journey of a protein begins with its primary structure, a simple linear sequence of amino acids linked by peptide bonds. Imagine that each amino acid is like a bead on a necklace, each contributing unique properties to the overall piece. The secondary structure arises when these chains begin to fold into patterns, often forming alpha helices and beta sheets. This folding is driven by interactions between the amino acids, such as hydrogen bonds, which are as delicate yet powerful as whispers in the wind. As we progress to the tertiary structure, the protein undergoes further folding, creating a three-dimensional shape that is essential for its function. This process can be likened to a sculptor carefully chiseling away at a block of marble, revealing the masterpiece within. Each fold and turn in the protein's structure can expose active sites or binding pockets, ready to interact with other molecules. In some cases, proteins are not complete until they form quaternary structures, where multiple polypeptide chains come together to function as a single unit. This cooperative behavior is beautifully illustrated by hemoglobin, the protein responsible for transporting oxygen in our blood. Hemoglobin consists of 4 subunits, and their cooperative binding allows for efficient oxygen transport, a vital aspect of our survival. As the eminent biochemist Max Perutz once stated, the structure of a protein is intimately related to its function. The mechanisms of protein synthesis and folding are equally captivating. The process begins in the nucleus of a cell, where the recipe for each protein is encoded in DNA. This blueprint is transcribed into messenger RNA, a single-stranded molecule that carries the instructions from the nucleus to the ribosomes, the cellular factories where proteins are assembled. As the ribosomes read the mRNA, they facilitate the translation of the genetic code into a polypeptide chain, linking amino acids in the precise order dictated by the mRNA. This journey from DNA to protein is an elegant ballet of molecular machinery, a dance of nucleotides and amino acids that brings life to the genetic code. But what happens after this chain is formed? The newly synthesized polypeptide must fold into its functional shape, a process that is not always straightforward. Chaperone proteins often come to the rescue, assisting in the folding process and ensuring that proteins achieve their correct structures. Without these helpers, misfolded proteins can lead to catastrophic consequences, such as diseases like Alzheimer's or cystic fibrosis. Proteins serve not only as structural components, but also as enzymes, the catalysts that accelerate chemical reactions within our bodies. Enzymes are remarkable in their specificity. Each type catalyzes a particular reaction, reducing the energy barrier and allowing reactions to occur at a pace suitable for life. Consider the enzyme amylase, found in our saliva, which begins the process of breaking down starches into sugars as we chew our food. This illustrates the seamless integration of proteins in our daily lives, highlighting their role not just in digestion, but in metabolism as a whole. Enzymatic functions can be likened to keys unlocking the doors of biochemical pathways. Each key, unique in its shape and structure, fits precisely into its corresponding lock, facilitating a cascade of reactions that ultimately sustain life. As the celebrated scientist Linus Pauling once remarked, "The greatest asset of a scientist is the ability to make connections. These connections manifest in the form of metabolic pathways, intricate networks of reactions that transform nutrients into energy and building blocks for cellular maintenance. Proteins play a pivotal role in these pathways, ensuring that energy flows efficiently and that cells have the necessary components for growth and repair." Take, for example, the enzyme lactase, which breaks down lactose into glucose and galactose, allowing our bodies to utilize the energy from dairy products. In some individuals, this enzyme diminishes with age, leading to lactose intolerance. This simple example illustrates how enzymes influence our interactions with food and can even shape cultural dietary practices across generations. Structural proteins, such as collagen and keratin, provide support and strength to various tissues in our bodies. Collagen, the most abundant protein in mammals, forms the scaffolding for skin, tendons, and ligaments, while keratin is found in our hair and nails, providing resilience and protection. These proteins are not only functional, but also aesthetic, contributing to the intricate beauty of living organisms. Imagine the delicate strands of collagen weaving through the layers of your skin, providing both structure and elasticity, allowing for movement and expression. It is within these proteins that we find a narrative of resilience and adaptability, showcasing nature's ability to craft materials that endure the test of time. Functional proteins extend beyond enzymes and structural components. Antibodies, produced by our immune system, are specialized proteins that recognize and neutralize foreign invaders, such as bacteria and viruses. Their ability to adapt and remember past encounters is a cornerstone of our immune defense, illustrating the remarkable versatility of proteins in protecting our health. Consider the words of immunologist Paul Ehrlich, who famously said, "The future belongs to those who see possibilities before they become obvious." This foresight is embodied in the evolution of proteins, which have adapted over millions of years to meet the needs of organisms in ever-changing environments. As we contemplate the vast landscape of proteins, it becomes clear that they are not merely the workhorses of cells, they are the architects of life itself. Each protein, with its distinct role and function, contributes to the symphony of existence, ensuring that every organism can thrive in its unique habitat. The intricate web of interactions among proteins reminds us of life's interconnectedness, where every action reverberates through the web of life, sustaining the delicate balance upon which all living beings depend. As we conclude our exploration of proteins and their essential roles in sustaining life, it becomes evident that these remarkable molecules are just one part of a much larger story. They work tirelessly within the intricate networks of our cells, yet they are only half of the narrative that defines cellular architecture and function. In the next chapter, we will delve into the world of lipids, the vital building blocks of membranes that not only provide structural integrity, but also play crucial roles in energy storage and cellular communication. Just as proteins orchestrate the biochemical symphony of life, lipids set the stage for this dynamic interplay, shaping the boundaries and interactions of every cell. Prepare to uncover how these versatile molecules contribute to the very essence of cellular existence and health. Chapter 4. Lipids, building blocks of membranes. Lipids, often seen as simple fats, are, in truth, remarkable molecules that serve as foundational building blocks of life. They come in various forms, each with its unique properties and purposes. Fats, phospholipids, and steroids are the three primary types of lipids that play crucial roles in the biochemistry of living organisms. Fats, also known as triglycerides, are composed of glycerol and 3 fatty acids. These molecules store energy in a dense form, providing a rich source of fuel for the body. Imagine a treasure chest filled with golden coins, each representing a calorie of energy, ready to be accessed when needed. This stored energy is essential, particularly during periods of fasting or prolonged exercise when immediate energy sources may be insufficient. Phospholipids, on the other hand, are the architects of cellular membranes. Their unique structure, with a hydrophilic head and 2 hydrophobic tails, allows them to form a bilayer that serves as a barrier, protecting the contents of the cell while regulating what enters and exits. Picture a serene lake, its surface smooth and undisturbed, concealing a vibrant ecosystem beneath. This bilayer not only provides structural integrity, but also creates a dynamic environment for the proteins embedded within, which are essential for various cellular functions. Steroids, such as cholesterol, add another layer of complexity to the role of lipids. Cholesterol, often vilified in popular culture, is vital for maintaining membrane fluidity. It can be likened to the lubricant in machinery, ensuring that the cellular machinery runs smoothly. Without it, membranes would become too rigid, impairing their function. As the esteemed biochemist Dr. R.A. Lazardi once said, "Cholesterol is not the enemy. It is a crucial player in the symphony of life." The role of lipids in cell membrane structure and function cannot be overstated. The cell membrane acts as a selective barrier, a gatekeeper that allows essential nutrients to flow in while keeping harmful substances at bay. This selective permeability is vital for maintaining homeostasis within the cell. As you listen, imagine a bustling city with traffic lights carefully regulating the flow of vehicles, ensuring that everything runs harmoniously. Moreover, lipid membranes are not static. They are fluid, allowing proteins to move within them, facilitating communication and interactions essential for cellular function. Think of a lively marketplace where vendors exchange goods and ideas, each stall representing a different protein interacting with others to create a thriving community. In addition to their structural roles, lipids are also vital for energy storage and insulation. Stored in adipose tissue, fats provide a reserve of energy that can be mobilized when required. This energy reserve is particularly important during periods of calorie restriction, when the body turns to its fat stores for sustenance. Visualize a bear preparing for hibernation, accumulating fat to sustain it through the long winter months. Insulation is another critical function of lipids, particularly in endothermic animals. Fatty tissues serve as thermal barriers, retaining heat and helping organisms maintain a stable internal temperature. Imagine a cozy blanket wrapped around you on a chilly night, providing warmth and comfort. This layer of fat not only protects against the cold, but also cushions vital organs, safeguarding them from injury. Lipid signaling adds yet another dimension to the role of these molecules in health and disease. Lipids act as signaling molecules, influencing various physiological processes, including inflammation, immune response, and metabolism. The discovery of lipid mediators has transformed our understanding of cellular communication. These molecules are like messengers carrying important information, sending signals that can initiate a cascade of responses within the cell. Consider the work of renowned biochemist Dr. A.W. Henson, who noted, "Lipids are the unsung heroes of cellular communication. They play a crucial role in how cells respond to hormonal signals, adapt to environmental changes, and manage stress." Imagine a phone ringing in a quiet room, drawing attention and prompting action. In this way, lipid signals can alert cells to respond to various stimuli, maintaining balance within the organism. The implications of lipid signaling in health are profound. Dysregulation of lipid metabolism is associated with various diseases, including obesity, diabetes, and cardiovascular disorders. As we reflect on this, it becomes clear that maintaining a balanced lipid profile is essential for overall health. Picture the delicate balance of a well-tended garden. Each plant relies on specific nutrients to thrive, similar to how cells depend on the right types and amounts of lipids for optimal function. The interconnectedness of lipid metabolism with broader health outcomes highlights the importance of understanding these molecules deeply. As you listen, imagine a world where the principles of biochemistry guide our choices, where we can harness the power of lipids for wellness. By understanding the roles of these vital molecules, we empower ourselves to make informed decisions about our health, from dietary choices to lifestyle habits. In this intricate biosphere of life, lipids emerge not merely as passive components, but as active participants in the symphony of cellular function. Their roles in energy storage, insulation, membrane structure, and signaling weave a rich narrative of life's complexity. As we delve deeper into the world of lipids, let us marvel at the elegance of these molecules and their profound impact on our existence. The journey through the world of lipids reveals a remarkable diversity, particularly when we consider the three primary categories: fats, phospholipids, and steroids. Each category plays distinct, yet interconnected roles, shaping not just the architecture of cells, but also influencing the delicate balance of life itself. Fats, often referred to as triglycerides, serve as the body's primary energy reservoir. Picture them as tiny energy-rich droplets stored in adipose tissue, waiting patiently to be called upon. When the body requires energy, these fats break down through a process known as lipolysis, releasing fatty acids that travel through the bloodstream to fuel various tissues. This process is vital for maintaining energy levels during periods of fasting or intense physical activity. The words of a renowned biochemist resonate here. Life is a series of energy transformations, and fats are the storage vessels that ensure these transformations are efficient and effective. Imagine a hibernating bear, nestled in its den, relying solely on its fat reserves to sustain it through the long, cold months. This magnificent adaptation showcases the critical function of fats not only in energy storage, but also in survival. In addition to energy, fats provide insulation, acting as a protective layer against temperature fluctuations, much like a cozy blanket that wraps around the body, ensuring stability in varying climates. Transitioning to phospholipids, we encounter molecules that possess a unique dual nature. On one side, they are hydrophilic or water-attracting, while on the other, they are hydrophobic or water-repelling. This remarkable property allows them to form bilayers, the fundamental structure of cell membranes. Picture, if you will, a vast ocean where phospholipids align themselves, creating a protective barrier that separates the interior of the cell from its external environment. This bilayer not only provides a structural framework, but also serves as a dynamic platform for various proteins and other molecules to interact. As cells communicate and respond to their surroundings, the fluidity of the phospholipid bilayer becomes paramount. It allows proteins to drift and interact in a manner akin to boats bobbing on gentle waves. The significance of this fluidity cannot be understated. It facilitates the movement of ions, nutrients, and signals, ensuring that the life within the cell remains vibrant and responsive. The interplay of lipids in membrane dynamics brings to life the concept of cellular organization. Within this fluid mosaic model, proteins embedded in the phospholipid bilayer perform a multitude of tasks. Some act as channels, allowing specific ions to flow in and out, while others serve as receptors, transmitting signals from the outside world to the cell's interior. As the eminent cell biologist once said, membranes are the gatekeepers of life, orchestrating the flow of information and resources essential for cellular function. Now let us turn our attention to steroids, another fascinating class of lipids. These molecules, characterized by a four-ring structure, play crucial roles in various biological processes. Think of cholesterol, the most well-known steroid, which is not just a villain in heart health, but also an essential component of cell membranes. Cholesterol contributes to membrane fluidity, preventing the bilayer from becoming too rigid in colder temperatures, much like a skilled conductor ensuring that each musical note resonates in harmony. Steroids also serve as precursors to vital hormones, which regulate numerous physiological functions. Consider cortisol, the stress hormone that helps the body respond to challenging situations, or testosterone and estrogen, which play significant roles in sexual development and reproductive health. The intricate balance of these hormones, derived from steroids, underscores the delicate interplay between lipids and bodily functions. In the words of a pioneering endocrinologist, hormones are the messengers of our bodies, and their synthesis begins with the very lipids that form the building blocks of life. As we explore further, we encounter the notion of lipid signaling, a complex yet fascinating aspect of lipid biology. Beyond their structural and energy storage roles, lipids act as signaling molecules that modulate physiological processes, influencing everything from inflammation to cell growth. Imagine them as tiny messengers, darting through the bloodstream, delivering vital information to cells throughout the body. For instance, certain phospholipids can be converted into signaling molecules known as eicosanoids, which play a pivotal role in inflammatory responses. These lipid-derived compounds can either promote or inhibit inflammation, depending on the body's needs. This duality highlights the importance of maintaining a proper balance in lipid profiles. It raises the question, how do we ensure that our bodies produce the right types of lipids at the right times? Diet plays a crucial role in this delicate balance. A diet rich in omega-3 fatty acids, found in fish and flax seeds, can promote the production of anti-inflammatory eicosanoids. In contrast, excessive consumption of saturated fats may lead to the production of pro-inflammatory lipids, contributing to chronic health conditions. As we reflect on this, it becomes apparent how our choices resonate through the intricate web of lipid metabolism, impacting not only our health but also the very fabric of our biological interactions. Imagining a world where we harness the insights from lipid research can inspire a transformative approach to health and wellness. By understanding the nuances of lipid metabolism, we can make informed dietary decisions, embracing foods that nourish our bodies and promote balance. The interconnectedness of lipids with health outcomes invites us to consider not just what we consume, but how these choices affect our overall well-being. The narrative of lipids is far from simple. It is a rich and intricate story that weaves through the very core of life. Their roles in energy storage, membrane structure, and signaling illuminate the complexity of biological systems. As we continue to explore this realm, let us remain curious about the myriad ways in which lipids shape our existence, fueling the essence of life itself. The journey through the lipid landscape is a testament to the elegance and intricacy of biology, and as we delve deeper, we uncover the profound impact these molecules have on our health and the world around us. As we conclude our exploration of lipids and their multifaceted roles in health and cellular function, it becomes clear that these molecules are not just passive players, but active participants in the intricate dance of life. Their influence extends beyond mere structure and energy, inviting us to appreciate the dynamic interplay that governs our biological systems. With this understanding, we can now turn our focus to the very blueprint of life, nucleic acids. In the next chapter, we will delve into the fascinating world of DNA and RNA, uncovering the mechanisms of replication, transcription, and translation that underpin genetic expression and heredity. Join us as we unravel how these molecular architects shape not only individual organisms, but also the evolution of life itself. Chapter 5: Nucleic Acids, the Blueprint of Life. In the intricate world of life, nucleic acids stand as the fundamental blueprint. These remarkable molecules, DNA and RNA, are the very essence of heredity, the carriers of genetic information that dictate the myriad forms of life on our planet. Imagine these strands, twisting and turning, like delicate ribbons of information, each twist a testament to the complexity of life itself. DNA, or deoxyribonucleic acid, resides primarily within the nucleus of eukaryotic cells, coiled and protected within chromatin. Its structure, resembling a double helix, features two strands intertwined, held together by complementary base pairs. Each base, adenine, thymine, cytosine, and guanine, forms a specific pairing that underpins the stability and integrity of genetic information. As James Watson famously remarked, it is the structure of the DNA molecule that makes the replication of genetic information possible. This elegant design is not merely aesthetic. It is functional, allowing for the precise storage and transmission of genetic material. RNA, or ribonucleic acid, plays a crucial role in the expression of this genetic blueprint. Unlike DNA, RNA is typically single-stranded and contains uracil instead of thymine. This seemingly small difference carries profound implications. RNA's structure allows it to fold into complex shapes, enabling it to perform various functions within the cell, from messenger RNA transporting genetic instructions to ribosomal RNA forming the core of ribosomes. Imagine the dance of these molecules as they work harmoniously, a symphony of biological activity that orchestrates life. The processes of replication, transcription, and translation are vital for the continuity of genetic information. During replication, the DNA helix unwinds, each strand serving as a template for the creation of a new complementary strand. This process ensures that each daughter cell inherits an identical copy of genetic material, a miracle of precision and fidelity. Transcription follows, where the information encoded in DNA is transcribed into messenger RNA. This process occurs in the nucleus, where RNA polymerase navigates the DNA template, synthesizing a strand of RNA that mirrors the sequence of the gene being expressed. As the renowned molecular biologist Sidney Brenner stated, the discovery of the role of messenger RNA was the key that unlocked the genetic code. This key, once turned, opens the door to the next stage, translation. Translation occurs in the cytoplasm, where ribosomes read the sequence of messenger RNA and translate it into a specific sequence of amino acids, forming proteins. This intricate machinery relies on transfer RNA, which brings the appropriate amino acids to the ribosome, allowing the assembly of proteins that perform countless functions within the cell. Imagine the precision of this process, akin to a chef following a recipe, each ingredient carefully measured and added to create a delicious dish. At the heart of this biological orchestra lies genetic coding and expression, the transition from genes to proteins. The genetic code is composed of triplets of nucleotides known as codons, each corresponding to a specific amino acid. This code is universal, shared by nearly all living organisms, a testament to the interconnectedness of life. As Francis Crick eloquently said, the central dogma of molecular biology is that information flows from DNA to RNA to protein. This flow is not merely a sequence of events, but a vital process that shapes the very fabric of life. The role of nucleic acids extends beyond individual organisms. They are central to heredity and evolution. Nucleic acids carry the information necessary for reproduction, ensuring that traits are passed from one generation to the next. Each organism's unique genetic makeup is a result of countless mutations and recombinations, shaping the diversity of life. Imagine the vast expanse of evolutionary history, where each change in nucleic acid sequences has contributed to the rich tapestry of life we observe today. As we ponder the significance of nucleic acids, we may ask ourselves, how do these molecules influence the future of life on Earth? The answer lies in their ability to adapt and evolve. Through mechanisms such as natural selection, organisms with advantageous traits are more likely to survive and reproduce, passing these traits along to future generations. This process is a delicate balance, guided by the principles encoded within nucleic acids. In understanding nucleic acids, we gain insight into the very essence of life itself. They are not merely molecules. They are the storytellers of our biological history, the architects of our potential. As we delve deeper into the role of nucleic acids, we uncover the profound connections that bind all living beings, a reminder of our shared journey through the cosmos. This journey, rich with discovery, beckons us to explore the mysteries that lie ahead. As we explore the intricate world of nucleic acids, we find ourselves drawn into the mesmerizing processes that underpin life itself. At the heart of these processes lies the structure and function of DNA and RNA, the primary players in the grand symphony of biological activity. DNA, with its iconic double helix structure, resembles a twisted ladder, where the rungs are made of pairs of nucleotides, each consisting of a sugar, a phosphate group, and a nitrogenous base. These bases, adenine, thymine, cytosine, and guanine, pair in a specific manner, creating a code that is vital for life. Imagine the precision with which these bases align, forming sequences that dictate everything from the color of a flower to the shape of a beak. As James Watson, one of the co-discoverers of the DNA structure, once remarked, "The secret of life is in the sequence." The sequence of these nucleotides encodes the instructions for building proteins, the workhorses of the cell, and ultimately, the entire organism. RNA, on the other hand, serves as a multifaceted messenger in the cell. Unlike the stable and durable DNA, RNA is often single-stranded, allowing it to fold and take on unique shapes. This versatility is essential for its roles in transcription and translation. During transcription, a segment of DNA unwinds, and RNA polymerase synthesizes a complementary strand of RNA. Picture this delicate dance, where the RNA strand emerges as a mirror image of the DNA template, carrying the genetic message from the nucleus to the cytoplasm. Once in the cytoplasm, the process of translation begins. Here, ribosomes read the RNA sequence, translating it into a specific sequence of amino acids, ultimately forming proteins. This remarkable transformation from nucleic acid to protein is akin to a musician interpreting a musical score, bringing notes to life through sound. The significance of this process cannot be overstated, as proteins perform critical functions, from catalyzing biochemical reactions to providing structural support. As we delve deeper into these processes, we might wonder, how is it that such intricate machinery functions so seamlessly? The answer lies in the intricate networks of regulatory mechanisms that govern gene expression. These mechanisms determine when and where genes are activated, ensuring proteins are produced in the right amounts and at the right times. Epigenetic modifications, such as methylation and acetylation, play a vital role in this regulation, acting like switches that can turn genes on or off without altering the underlying DNA sequence. Consider the profound implications of these regulatory systems. They allow organisms to adapt to their environments and respond to various stimuli. For instance, the ability of a plant to grow toward sunlight, or a rabbit to develop a thicker coat in winter, illustrates the dynamic interplay between genetic information and environmental cues. As Charles Darwin eloquently stated, "It is not the strongest of the species that survive, nor the most intelligent, but the one most responsive to change." The story of nucleic acids does not end with individual organisms. It extends into the realms of heredity and evolution. Each generation passes down a portion of its genetic material, creating a lineage that carries the legacy of adaptations and traits. This transmission of information ensures that life is not static, but is instead a continuous journey of change and evolution. Imagine the vast tree of life, where branches represent different species, each one a testament to the countless iterations of genetic material that have been passed down through the ages. From the simplest bacteria to the most complex mammals, Each organism carries a unique narrative encoded within its nucleic acids. As we reflect on this narrative, we may ask ourselves, how do these stories influence the future of life on Earth? The answer lies in the ongoing processes of mutation and natural selection. Mutations, spontaneous changes in the DNA sequence, serve as the raw material for evolution. Some mutations confer advantages, while others may be neutral or even detrimental. Those that enhance survival and reproductive success are more likely to be passed on, shaping the genetic landscape of populations over time. This process of evolution is not merely a historical account. It is an active, vibrant phenomenon that continues to unfold each day. Consider the example of antibiotic resistance in bacteria. As we expose these microorganisms to antibiotics, those with mutations that confer resistance survive and reproduce, leading to a population that is increasingly difficult to treat. This real-time evolution serves as a poignant reminder of the power of nucleic acids in shaping life. As we immerse ourselves in the study of nucleic acids, we begin to appreciate their role not only in the mechanics of life, but also in the interconnectedness of all living beings. The shared genetic heritage that binds us together, from the smallest microbes to the largest mammals, is a testament to the unity of life on this planet. In light of this, we can ponder the ethical and philosophical implications of our understanding of nucleic acids. As we delve into the realms of genetic engineering and synthetic biology, we stand at a crossroads, where the power to manipulate life itself raises profound questions about the boundaries of science and morality. What does it mean to alter the blueprint of life? How do we balance innovation and responsibility? These questions invite us to reflect on our role as stewards of the natural world, emphasizing the importance of understanding the intricacies of life at a molecular level. As we continue to explore the depths of nucleic acids, we uncover more than just the mechanics of biology. We encounter the threads that weave our existence together. Each strand of DNA, each molecule of RNA, encapsulates the stories of our past, the complexities of our present, and the possibilities of our future. In this journey through the landscape of nucleic acids, we are reminded that we are all part of a larger narrative, one that transcends individual experiences and connects us to the very fabric of life itself. The exploration of these molecules is not merely an academic pursuit. It is an invitation to embrace the wonder of existence and the profound mysteries that lie ahead. As we stand on the precipice of discovery, we can feel the heartbeat of life resonating within us, urging us to delve deeper into the enigma of nucleic acids and the remarkable potential they hold for the future of all living beings. As we reflect on the profound influence of nucleic acids in shaping life's intricate tapestry, we are reminded of the fundamental processes that allow organisms to thrive and adapt. The stories encoded within our DNA not only narrate the past, but also set the stage for the dynamic interplay of energy and matter that sustains life. This journey through the genetic blueprint naturally leads us to explore metabolism, the essential mechanism by which living organisms convert resources into energy. From the elegant dance of catabolic and anabolic pathways to the pivotal role of ATP as the energy currency of cells, the next chapter invites us to uncover how life continuously transforms and harnesses energy in myriad ways. As we transition into this exploration of metabolic processes, we will uncover the vital connections that enable the symphony of life to flourish. Chapter 6. Metabolism: Transforming Energy in Living Organisms. In the realm of living organisms, metabolism serves as the transformative force, a complex orchestra that plays out within every cell. This intricate process is not merely about the breakdown of substances, but also about the construction of life itself. At the heart of metabolism lies a powerful duo, catabolism and anabolism. Catabolism refers to the pathways that break down molecules to release energy, while anabolism encompasses the pathways that build up compounds, utilizing energy to create the structures necessary for life. Imagine a bustling city where some workers are focused on demolishing old structures to make way for new developments. These workers symbolize catabolic pathways, breaking down carbohydrates, fats, and proteins into simpler molecules, releasing energy in the process. This energy is captured in the form of adenosine triphosphate, or ATP, the energy currency of the cell. ATP can be envisioned as a rechargeable battery, storing energy in its high-energy phosphate bonds. When cells require energy for various tasks, be it muscle contraction, nerve impulse propagation, or the synthesis of macromolecules, they rely on ATP. The moment that a phosphate group is released from ATP, energy is liberated, ready to be harnessed for cellular activities. On the other side of the metabolic spectrum, we encounter anabolism, where the energy from ATP is utilized to construct essential biomolecules. This process is akin to artisans crafting intricate sculptures, molding raw materials into proteins, nucleic acids, and other vital compounds. An example of anabolic activity is the synthesis of proteins from amino acids, a process that not only builds cellular structures, but also facilitates the myriad functions that sustain life. As we delve deeper into the mechanisms of metabolism, we encounter cellular respiration. This vital process can be categorized into aerobic and anaerobic pathways. In aerobic respiration, which occurs in the presence of oxygen, glucose is fully oxidized, yielding carbon dioxide and water, along with a substantial amount of ATP. Picture a serene lake, where the clear waters reflect the sunlight, symbolizing the efficiency of aerobic respiration and its ability to harvest energy from glucose efficiently. Conversely, anaerobic respiration occurs in the absence of oxygen. It is here that life finds a way to adapt, showcasing the resilience of organisms. Through fermentation, certain bacteria and yeast can convert glucose into energy, producing byproducts such as ethanol or lactic acid. This process, while less efficient than aerobic respiration, allows cells to survive in environments where oxygen is scarce. Can you imagine a world where life persists in the dim recesses of a cave, thriving in darkness, yet still generating energy? A fascinating aspect of metabolism is its interplay with photosynthesis, the process by which plants capture light energy and convert it into chemical energy stored in glucose. This process unfolds in the chloroplasts, where sunlight is absorbed by chlorophyll, the green pigment that adorns the leaves. Imagine a sun-drenched field, each blade of grass dancing in the wind, soaking up sunlight, as the chloroplasts within their cells tirelessly convert light into energy. Photosynthesis occurs in two stages: the light-dependent reactions and the light-independent reactions, also known as the Calvin cycle. During the light-dependent reactions, sunlight energizes electrons, which then travel through a series of proteins embedded in the thylakoid membrane. This journey generates ATP and another energy carrier, NADPH, which are essential for the next phase of photosynthesis. The Calvin cycle takes place in the stroma of the chloroplast, where carbon dioxide is fixed into glucose using the energy harnessed from the light-dependent reactions. As the renowned biologist Albert Einstein once said, "Look deep into nature, and then you will understand everything better." This sentiment rings true when we observe the delicate balance of metabolic pathways that sustain life on Earth. Each organism, whether a towering redwood or a tiny bacterium, engages in these processes, contributing to the vast interconnected web of life. Consider the implications of these metabolic processes. How do they affect our daily lives? The food we consume is transformed through catabolic pathways into the energy we need to thrive. The oxygen we breathe, a byproduct of photosynthesis, sustains our cellular respiration. In a sense, we are intimately connected to the metabolic processes of plants and other organisms, relying on them for our very existence. In exploring metabolism, we uncover not only the mechanics of energy transformation, but also the profound interconnectedness of life. Each pathway, whether catabolic or anabolic, plays a crucial role in the grand design of biological systems. As we ponder the intricate relationships within ecosystems, it becomes clear that metabolism is not just a series of reactions, but an eloquent narrative of survival, adaptation, and continuity. As we reflect on the wonder of these processes, we may find ourselves feeling a sense of calm and connection to the natural world. The rhythm of metabolism mirrors the ebb and flow of life itself, reminding us of the delicate balance that sustains all living beings. Embracing this knowledge can provide a soothing perspective, a reminder of the intricate beauty that exists both within and around us. In the heart of every living cell lies a remarkable molecule known as adenosine triphosphate, or ATP. This tiny powerhouse is often referred to as the energy currency of the cell, a phrase that encapsulates its vital role in fueling the multitude of processes that sustain life. Imagine, if you will, a bustling marketplace where energy is exchanged as currency. ATP acts as the coin that facilitates transactions between various cellular activities. ATP is synthesized through both catabolic and anabolic pathways, a dynamic interplay where energy is both released and stored. When glucose or fatty acids are broken down during cellular respiration, energy is released, and a portion of that energy is harnessed to synthesize ATP. This process is akin to harnessing the wind to turn a mill, transforming raw energy into a usable form. As Albert Einstein once said, "Energy cannot be created or destroyed, it can only be transformed." In this sense, ATP serves as a bridge, connecting the raw energy of food to the vital processes that keep us alive. Now let us delve deeper into the process of cellular respiration itself, which exists in two forms, aerobic and anaerobic. Aerobic respiration, as the name suggests, requires oxygen and takes place in the mitochondria of eukaryotic cells. Picture the mitochondria as tiny power plants, efficiently converting fuel into energy. During this process, glucose reacts with oxygen, producing carbon dioxide and water as byproducts, all while releasing a wealth of energy that is then captured as ATP. The elegance of aerobic respiration is not only in its efficiency, but also in its ability to sustain complex life forms, allowing them to thrive in diverse environments. In contrast, anaerobic respiration occurs in the absence of oxygen. This process is often less efficient but equally fascinating. For instance, consider yeast, a microorganism that has been utilized by humans for thousands of years in the production of bread and beer. Under anaerobic conditions, yeast ferments sugars, producing alcohol and carbon dioxide. This fermentation process has been pivotal in human culture, underpinning the art of brewing and baking. As the biologist Louis Pasteur observed, "Wine is sunlight, held together by water." This phrase beautifully captures the transformation of energy, from sunlight to sugar to alcohol, demonstrating the intricate pathways of life that intertwine biology and culture. But what happens in our bodies when oxygen is scarce? In such moments, our muscles also resort to anaerobic respiration, producing lactic acid as a byproduct. This can lead to that familiar sensation of fatigue during intense exercise. Have you ever wondered why this happens? The answer lies in the body's remarkable way of adapting to stress, allowing us to push our limits in the pursuit of strength and endurance. As we explore the role of metabolism in living organisms, we encounter the wondrous phenomenon of photosynthesis. This process is the cornerstone of life on Earth, enabling plants, algae, and certain bacteria to convert light energy into chemical energy. Picture a sunlit leaf capturing sunlight and transforming it into the building blocks of life. The chlorophyll pigments within plant cells absorb light, initiating a series of reactions that ultimately convert carbon dioxide and water into glucose and oxygen. In this way, plants serve as the foundation of the terrestrial food web, supporting not only themselves but also the myriad of organisms that depend on them. The significance of photosynthesis cannot be overstated. As the biochemist Melvin Calvin expressed, photosynthesis is the process by which the sun's energy is converted into chemical energy, the energy of life. This conversion is not merely a scientific concept, it is the very essence of life itself, allowing energy to flow through ecosystems. Imagine the intricate connections between plants and animals, each playing their role in a grand symphony of life, all made possible by this remarkable process. Yet the story of metabolism does not end here. It is essential to recognize that metabolic pathways are not isolated. They are interconnected, forming a complex network that regulates energy production and utilization. For instance, the glucose produced during photosynthesis can be stored as starch in plants, a reservoir of energy that can be tapped into when needed. When animals consume these plants, they enter a cycle of energy transfer, illustrating the interconnectedness of life. Metabolism also adjusts to the needs of the organism. During periods of abundance, anabolic pathways flourish, synthesizing complex molecules from simpler ones, building up reserves for future use. Conversely, in times of scarcity, catabolic pathways dominate, breaking down stored nutrients to release energy. This delicate balance is a testament to the adaptability of life, a reminder that survival often hinges on the ability to respond to changing conditions. As we reflect on metabolism's intricate dance, one cannot help but marvel at the outstanding individuals who have contributed to our understanding of these processes. Scientists such as Hans Krebs, who elucidated the Krebs cycle, have paved the way for our comprehension of cellular respiration. His work, which revealed how living organisms convert food into energy, was monumental. Krebs once stated, "The living cell is a microcosm of the universe, indicating the profound complexity and elegance of life at the cellular level." In contemplating these metabolic pathways, it becomes evident that they are not just mechanisms of energy transformation, they are narratives that speak to the essence of life itself. Through catabolism and anabolism, through respiration and photosynthesis, organisms engage in a continuous dialogue with their environment, adapting and thriving in a world that is ever-changing. Consider how these processes resonate within you. Every breath you take, every bite of food you consume, connects you to this grand narrative. The energy that fuels your thoughts, your movements, and your very existence is derived from the same intricate pathways that have sustained life for eons. In this shared experience, we find a sense of unity with all living beings, a reminder that the rhythm of metabolism echoes through the ages, linking us to the past, present, and future of life on Earth. As you reflect on this interconnectedness, allow yourself to feel a gentle calm wash over you, a soothing acknowledgement of the beauty that lies within these processes. Even in the quiet moments, when the world around you slows, the vibrant dance of metabolism continues, a testament to the resilience and adaptability of life in all its forms. As we delve deeper into the narratives of life, we must now turn our attention to the unsung heroes of metabolism, enzymes. These remarkable catalysts play a pivotal role in facilitating the intricate reactions that sustain biological processes. Understanding their structure and function will illuminate how they drive the very transformations that fuel energy flow within living organisms. In the next chapter, we will explore the mechanisms by which enzymes operate, the factors that influence their activity, and their profound impact not only on biology but also on biotechnology and industry. Join us as we uncover the dynamic world of enzymes, where every interaction is a vital piece of the broader tapestry of life. Chapter 7. Enzymes, catalysts of biological reactions. Enzymes are remarkable biological catalysts that orchestrate the myriad of metabolic reactions within living organisms. They play an essential role in facilitating chemical transformations, enabling life to thrive in the complex interplay of biochemistry. Without enzymes, the countless reactions necessary for survival would occur far too slowly to sustain life, making these molecules indispensable. The structure and function of enzymes are intricately linked. Each enzyme possesses a unique three-dimensional shape, which includes a specific region known as the active site. This active site is where the substrate, or the molecule upon which the enzyme acts, binds. Imagine a key fitting into a lock. The enzyme's active site is the lock, and the substrate is the key. When the substrate enters the active site, the enzyme undergoes a subtle change, allowing the chemical reaction to proceed more efficiently. This phenomenon is often described as the induced fit model, where the enzyme adjusts its shape to better accommodate the substrate. The mechanisms of enzyme activity are fascinating. Enzymes can lower the activation energy required for a reaction, allowing it to proceed at a remarkable pace. This acceleration is achieved through various means. Some enzymes stabilize the transition state of the substrate, while others may provide an environment conducive to the reaction, such as a hydrophilic pocket that facilitates the movement of ions. The specificity of enzymes ensures that each reaction is finely tuned, often described by the saying, lock and key, or induced fit. The elegance of this specificity is evident in nature's design, where each enzyme interacts with only certain substrates, ensuring that metabolic pathways proceed smoothly. Regulation of enzyme activity is crucial for maintaining homeostasis within the cell. Enzymes can be activated or inhibited by various factors, allowing the organism to respond to changes in its environment or internal state. For instance, allosteric regulation involves molecules binding to sites other than the active site, inducing conformational changes that either enhance or diminish enzyme activity. This regulatory feature enables cells to adapt quickly to fluctuating conditions, ensuring that metabolic processes align with the organism's needs. Several factors can significantly affect enzyme activity, including temperature, pH, and substrate concentration. Enzymes typically have an optimal temperature range, within which they function most efficiently. At temperatures that are too low, the kinetic energy of molecules decreases, slowing down the reaction rates. Conversely, at excessively high temperatures, enzymes may denature, losing their structure and functionality. The pH level is another critical factor. Each enzyme has an optimal pH at which its activity is maximized. For example, pepsin, an enzyme found in the stomach, thrives in the acidic environment, while others, like trypsin, prefer a more neutral pH found in the small intestine. Substrate concentration also plays a pivotal role in enzyme activity. As the concentration of substrate increases, the rate of reaction typically rises until a saturation point is reached, where all active sites are occupied. This concept can be visualized as a busy highway. As more cars enter, traffic increases until the road is fully packed, causing a bottleneck where no additional cars can fit. Enzymes have found their way into various applications in biotechnology and industry, transforming processes and enhancing productivity. In the realm of medicine, enzymes are harnessed for diagnostics and therapies. For example, enzymes like lactase are used to break down lactose in products designed for lactose-intolerant individuals, allowing them to enjoy dairy without discomfort. In the food industry, enzymes are employed in brewing, baking, and cheese-making, enhancing flavors and textures. The textile industry also utilizes enzymes to improve fabric treatment, offering a more sustainable alternative to harsh chemicals. The field of biocatalysis, which leverages enzymes to facilitate chemical reactions, is growing rapidly. Enzymes can catalyze reactions under mild conditions, reducing the energy requirements and minimizing waste. This efficiency not only serves the environment, but also paves the way for more cost-effective processes in manufacturing. As we delve deeper into the world of enzymes, we can appreciate their intricate roles in life. They are not merely chemical facilitators. They embody the elegance and complexity inherent in biological systems. Each enzyme is a testament to nature's ingenuity, crafting a delicate balance that sustains life. The beauty of enzymes lies in their specificity, their ability to catalyze reactions with remarkable precision, and their capacity to adapt to the ever-changing landscape of biological needs. The next time you consider the metabolism within your own body, think of the countless enzymes at work, tirelessly ensuring that every reaction unfolds just as it should. The intricate dance of these molecules is a reminder of the delicate balance that sustains life, a balance that is constantly in motion, adapting and responding to the world around us. What a profound realization it is that within each of us there exists a bustling world of enzymes, driving the very essence of our being. As we explore the structure of enzymes, we find that each enzyme is a marvel of molecular architecture. At the heart of an enzyme's function lies its active site, a unique region where substrates, the molecules upon which enzymes act, bind. Imagine a lock and key. The active site is the lock, specifically shaped to fit only a particular key, the substrate. This specificity is crucial, as it ensures that enzymes catalyze only certain reactions, thus maintaining the delicate equilibrium of metabolic pathways. The interaction between an enzyme and its substrate is not just a simple embrace, it is a dynamic relationship. When a substrate binds to the active site, it induces a change in the enzyme structure, a phenomenon known as the induced fit model. This adjustment allows the enzyme to better catalyze the reaction, lowering the activation energy required for the reaction to occur. This elegant mechanism underlies the efficiency of enzymes and highlights their role as biological catalysts. Consider the words of the renowned biochemist Michael Behe, who once said, "Biochemistry is the study of life at the molecular level." Through the lens of enzymes, we can truly appreciate this statement. They are not merely catalysts. They are the architects of life's intricate processes, orchestrating reactions that allow cells to function, grow, and thrive. Enzyme activity is not static. It is a finely tuned symphony influenced by various factors. Temperature plays a significant role in enzyme regulation. Each enzyme has an optimal temperature range at which it operates most efficiently. As temperatures rise, enzyme activity generally increases, leading to faster reactions. However, this is a double-edged sword. Excessive heat can denature enzymes, altering their structure and rendering them inactive. Imagine the delicate balance of a warm summer day, where a gentle breeze enhances comfort, but too much heat can lead to discomfort or harm. Similarly, pH levels significantly affect enzyme function. Different enzymes thrive in specific pH environments. For instance, pepsin, an enzyme in the stomach, is adapted to function optimally at the acidic pH of gastric juice, while others, like amylase found in saliva, work best in a more neutral environment. This diversity in pH adaptation underscores the intricate relationships enzymes have with their surroundings. As the celebrated molecular biologist Peter Agary remarked, "The truth is, we are all a part of the same universe. We are all interrelated." So too are enzymes interrelated with their environments, adapting to fulfill their roles. Concentration of substrates also influences enzyme activity. An increase in substrate concentration often leads to an increase in reaction rate, up to a certain point. Beyond that threshold, the enzyme becomes saturated, and the reaction rate levels off as all active sites are occupied. Picture a bustling marketplace. As more shoppers arrive, the energy and activities increase until the space becomes too crowded, and movement slows down. This saturation point illustrates the finite capacity of enzymes to engage with substrates, emphasizing the need for balance in biochemical processes. Enzymes not only govern reactions within living organisms, they are also pivotal in biotechnology and industrial applications. Their ability to function under mild conditions and with high specificity makes them invaluable in various fields. In the realm of medicine, enzymes are harnessed for diagnostic purposes. For instance, the enzyme lactase is used to diagnose lactose intolerance, providing a simple method for understanding digestive health. Imagine the relief felt by someone who discovers the root cause of their discomfort with the help of a single enzyme. This is the power of biochemistry at work. In the textile industry, enzymes such as cellulases are employed to enhance fabric quality. They help in biopolishing, which removes fuzz from fabrics, leading to a smoother finish. This enzymatic treatment not only improves the aesthetics of clothing, but also reduces the need for harsh chemicals, making the process more environmentally friendly. Picture a soft, beautifully finished garment, the result of nature's ingenuity harnessed through enzymes. In the brewing and baking industries, enzymes such as amylase and protease are essential for breaking down starches and proteins, respectively. These enzymes contribute to flavor development and texture, transforming simple ingredients into delightful creations. Imagine the rich aroma of freshly baked bread or the crispness of a well-crafted beer, both made possible by the unseen work of enzymes that enhance flavor and mouthfeel. The food industry also benefits from enzymes in food preservation. For example, lysozyme is used to combat unwanted bacteria in cheese production, while transglutaminase is utilized to improve texture and binding in processed meats. The application of enzymes in food processing not only enhances quality, but also promotes safety, allowing consumers to enjoy their meals with peace of mind. As we delve deeper into the world of enzymes, we find that their applications extend beyond conventional boundaries. In agriculture, enzymes are used to improve soil health and enhance nutrient availability for crops. Various biofertilizers incorporate enzymes that facilitate the breakdown of organic matter, enriching the soil and promoting sustainable farming practices. Imagine lush green fields, thriving plants nourished by the invisible hand of enzymes working diligently beneath the surface. Moreover, the ongoing research in enzyme engineering and synthetic biology promises exciting advancements. Scientists are developing enzymes with tailored properties for specific applications, paving the way for innovative solutions in energy production, waste management, and beyond. The potential of enzymes to contribute to a more sustainable future is immense. Each discovery adds another thread to the intricate web of life, revealing the boundless possibilities that await us. As we reflect on the remarkable journey of enzymes, we recognize their pivotal role as catalysts of biological reactions. They are the silent orchestrators of life, ensuring that each reaction unfolds with precision and grace. The next time you savor your favorite dish or enjoy a refreshing beverage, take a moment to appreciate the unseen forces at play. Enzymes are more than just molecules. They are the lifeblood of biological processes, the intricate links that connect the vast tapestry of life. With each reaction they facilitate, they remind us of the beauty and complexity of the world around us. A world shaped by the delicate balance of nature's design. What wonders lie ahead as we continue to explore this captivating realm? The journey of discovery is just beginning. As we marvel at the intricate roles enzymes play in transforming our world, we cannot overlook the interconnectedness of life that they help sustain. These catalysts not only enhance our culinary experiences, but also contribute to the health of ecosystems and innovative solutions for the future. Just as enzymes facilitate essential biochemical processes, so too do cells communicate through a myriad of signals that govern their behavior and interactions. This complex dialogue, driven by hormones, neurotransmitters, and pheromones, is crucial for maintaining balance within living organisms. With this understanding, we now turn our attention to the fascinating realm of cellular communication, where signaling pathways and responses shape the very fabric of life itself. What mechanisms will we uncover as we delve into this vital aspect of biological systems? Chapter 8. Cellular Communication, Signaling Pathways and Responses. In the intricate world of cellular communication, life unfolds through a symphony of signaling pathways, where each cell plays its unique note in the grand arrangement of existence. At the core of this communication lies the process of cellular respiration, a vital mechanism that transforms nutrients into energy. Imagine this transformation as a delicate alchemy, where the food we consume is broken down into its elemental parts, enabling the cells to harness energy in the form of adenosine triphosphate, or ATP. This energy currency fuels all cellular activities, from muscle contractions to the synthesis of vital molecules. As the nutrients are metabolized, they undergo a series of complex biochemical reactions. Glucose, for instance, is oxidized in a process that produces not only ATP, but also carbon dioxide and water. This process occurs in the mitochondria, often referred to as the powerhouse of the cell. It is here, in this organelle, that the magic of transforming the chemical energy of food into a form usable by the cell takes place. Each heartbeat, every breath, is a testament to this remarkable conversion, a reminder of the unseen work that sustains life. Cellular signals can be likened to whispers in a crowded room, carrying messages that direct cells to respond to their changing environment. These signals come in various forms, such as hormones, neurotransmitters, and pheromones. Hormones, produced by glands in the endocrine system, travel through the bloodstream to target organs. They regulate processes like growth, metabolism, and mood. When we feel stress, for instance, adrenaline surges through our body, preparing us for action. "The only thing we have to fear is fear itself," said Franklin D. Roosevelt, capturing the essence of how our body responds to signals of stress that can either paralyze or propel us into motion. Neurotransmitters, on the other hand, communicate messages across the synapses between nerve cells. They relay signals that affect everything from muscle contraction to emotional states. Imagine a neurotransmitter as a key that unlocks a door, allowing the passage of information. Dopamine, known as the feel-good neurotransmitter, plays a crucial role in reward and pleasure systems within the brain, guiding behaviors that promote survival. Pheromones serve a different purpose, acting as chemical signals released by one organism to influence the behavior of another. These signals can trigger profound responses, particularly in the animal kingdom, where they often govern mating rituals and territorial boundaries. As we ponder the significance of these subtle cues, one might ask, how much of our behavior is influenced by unseen signals? The answer lies in the profound interconnectedness of all living things, where each organism responds to the silent language of pheromones, shaping interactions and relationships within ecosystems. At the heart of cellular communication are receptors, specialized structures that receive and interpret these signals. Picture a receptor as a finely tuned instrument, designed to respond to specific frequencies of communication. When a signal molecule binds to its receptor, it initiates a cascade of events known as signal transduction pathways. This intricate process translates the external message into a cellular response, akin to converting sound waves into music. One of the most remarkable features of these pathways is their complexity and specificity. For instance, the binding of a hormone to its receptor activates a series of proteins within the cell, each playing a distinct role in amplifying and relaying the signal. This leads to a multitude of responses, from altering gene expression to changing cell metabolism. Each pathway is a carefully orchestrated sequence, reminiscent of a conductor guiding an orchestra, ensuring that each note contributes to the harmonious outcome of cellular function. The importance of feedback loops in maintaining homeostasis cannot be overstated. These loops are the regulatory mechanisms that keep our internal environment stable, despite external fluctuations. For example, when blood sugar levels rise after a meal, insulin is secreted to facilitate the uptake of glucose by cells. Once blood sugar levels return to normal, insulin secretion decreases, illustrating a classic negative feedback loop. This dynamic balance is essential for survival, as it allows organisms to adapt to changing conditions while preserving their core functions. Feedback mechanisms can also serve as positive loops, amplifying responses in certain situations. A prime example can be seen during childbirth, where the release of oxytocin enhances contractions, propelling the process forward until delivery is achieved. As the saying goes, life is a series of cycles, and these feedback loops reflect the cyclical nature of biological processes. In the realms of development and immunity, signaling pathways play pivotal roles, guiding the growth and differentiation of cells. During embryonic development, cells communicate through a series of signals that dictate their fate, whether they will become neurons, muscle cells, or blood cells. This intricate choreography of signaling is what allows a single fertilized egg to develop into a complex organism. Consider the influence of growth factors, proteins that stimulate cell proliferation and differentiation. They act as the architects of development, guiding cells to form tissues and organs. The discovery of these growth factors revolutionized our understanding of biology. As Dr. Rita Levi-Montalcini, a pioneer in this field, once said, "What is important to me is to keep the sense of wonder alive, reflecting the awe-inspiring nature of cellular communication in shaping life." Immune responses also hinge on signaling pathways, allowing cells to recognize and respond to pathogens. When a pathogen invades, it triggers a cascade of signals that mobilize immune cells to the site of infection. This intricate signaling network ensures that the body can respond swiftly and effectively to threats. Imagine a fortress where sentinels communicate through signals, alerting the rest of the kingdom to prepare for battle. The cooperation among immune cells is facilitated by cytokines, signaling molecules that modulate the immune response. They are the whispers in the immune system, guiding cells to either ramp up their defenses or to resolve inflammation once the threat has been neutralized. This balance is crucial for health, as an overactive immune response can lead to autoimmune diseases where the body mistakenly attacks its own tissues. As we reflect on the elegance of cellular communication, we begin to appreciate the profound interconnectedness of life. Each signal, each response, weaves a story of adaptation and survival, illustrating the remarkable capacities of living organisms. Can you imagine the complexity of these interactions, where countless signals converge and diverge, shaping the destiny of cells and, ultimately, the entire organism? In this intricate network, the balance of signals and responses is not just about survival, it is about thriving in an ever-changing world. As we delve deeper into the wonders of cellular communication, we uncover the threads that connect us all, inviting us to marvel at the beauty of life itself. Cellular communication is not merely a response to immediate dangers, but a finely tuned orchestra that also plays a vital role in development and growth. From the moment of fertilization, signaling pathways guide the transformation of a single cell into a complex organism, orchestrating the delicate balance between differentiation and proliferation. Imagine the early stages of life within the womb, where cells, through their intricate language of signals, decide their fates, whether to become a heart cell, a neuron, or a skin cell. Among these pivotal players are growth factors, proteins that act as key messengers in development. They bind to their specific receptors, igniting cascades of activity that tell cells to divide, migrate, or specialize. As developmental biologist Sir John Gurdon once stated, "The most important thing is that the cell has the ability to respond to its environment and to take on a form that is appropriate for its role." This responsiveness is a testament to the dynamic nature of cellular communication. Consider the process of limb development. In a developing embryo, a small region of cells known as the apical ectodermal ridge sends out signals that dictate the growth of fingers and toes. This ridge acts like a conductor, ensuring that the right signals are sent at the right time. The precision of these signals is crucial. A minor disruption can lead to malformations, emphasizing the delicate interplay of communication that guides life's intricate designs. As we ponder this journey from a single cell to a fully formed organism, we can almost feel the pulse of life, echoing through the corridors of development. Each phase of growth relies on feedback loops that fine-tune cellular responses, maintaining balance and harmony. These loops are the body's way of ensuring that signals do not run amok, but instead adjust according to the needs of the organism. Feedback mechanisms are essential in maintaining homeostasis, the state of steady internal conditions. Negative feedback loops act like a thermostat, where a signal triggers a response that dampens the initial signal, ensuring stability. For example, consider the role of insulin in glucose regulation. When blood sugar levels rise, insulin is secreted, prompting cells to absorb glucose and lower blood sugar levels. Once equilibrium is restored, insulin secretion decreases, demonstrating the elegance of this communication. Conversely, positive feedback loops amplify a response until a certain goal is achieved. The process of childbirth offers a vivid illustration. During labor, the hormone oxytocin is released, intensifying contractions. These contractions lead to more oxytocin release, creating a powerful cycle that drives the process forward. This interplay of signals showcases the dual nature of cellular communication, both stabilizing and propelling life's essential processes. The beauty of cellular signaling also extends into the realm of adaptation. Think of how organisms respond to changing environments, adjusting their internal signaling pathways to thrive in new conditions. For instance, plants exhibit remarkable abilities to respond to light and gravity through a process known as tropism. Cells communicate through hormones like auxins, which regulate growth direction based on environmental cues. Imagine a young sapling stretching toward the sun, its cells communicating tirelessly to ensure it turns its leaves to capture maximum light. In the animal kingdom, pheromones represent another fascinating aspect of signaling. These chemical messengers are released into the environment, conveying messages that can influence behavior, reproduction, and social dynamics. The ability of ants to follow pheromone trails back to their colony is a striking example of this form of communication. As biologist E.O. Wilson said, ants are a model for how to integrate behavior with ecology. Through such intricate signaling, they demonstrate the power of cooperation and community in the natural world. As we explore these pathways, we begin to appreciate the sophistication embedded within every cell. The intricate signaling mechanisms that allow for communication, adjustment, and response are not merely biological processes, they are the essence of life itself. Each signal is a note in a larger symphony, creating harmony that resonates through the veins of existence. Can you envision the constellations of signals that light up within each organism, guiding their journey through life? The delicate balance of these pathways illustrates a profound truth. Life is not a solitary endeavor, but a collective experience, where every cell, every signal, plays its part in the grand narrative of existence. The story of cellular communication is an invitation to marvel at the unseen connections that bind us all, a reminder of the intricate beauty that underlies the fabric of life. As we delve deeper into the realms of cellular signaling, we uncover the mysteries of how life continuously adapts, thrives, and evolves. Each discovery is like a whispered secret, revealing more about the interconnected web of existence. The journey through this cellular landscape is endless, inviting us to remain curious and open to the wonders of life that unfold around us. As we reflect on the intricate web of cellular communication, it becomes clear that these signaling pathways are not isolated phenomena. They are deeply influenced by the environment in which organisms exist. Just as a sapling reaches toward the sun, so too do cells respond dynamically to external factors that shape their biochemical reactions. This interplay between signaling and environmental influences invites us to explore how temperature, pH, and other conditions impact life at the molecular level. In the following chapter, we will delve into the role of these environmental factors, examining the remarkable adaptations of organisms to extreme conditions and the consequences of pollutants and climate change on biochemical processes. This journey promises to reveal further layers of complexity in the dance of life, as we uncover how the environment not only challenges, but also shapes the very essence of biochemistry. Chapter 9. The Role of Environmental Factors in Biochemistry. In the heart of our planet's ecosystems lies a remarkable process that fuels life itself: photosynthesis. Imagine the sun, a golden orb hanging in the vast sky, casting its warm rays upon the foliage. Leaves, with their intricate design, capture this energy, transforming it into chemical nourishment. Through the delicate interplay of chlorophyll, sunlight, carbon dioxide, and water, these green architects create glucose and oxygen, the very foundations of our food web. As the botanist John W. Harshberger once said, photosynthesis is the basis of life on Earth, encapsulating the essence of this vital process. As we delve deeper into the biochemical reactions that sustain life, we must consider the myriad of factors that influence these processes. Temperature, pH, and concentration all play pivotal roles in determining how efficiently reactions occur. For instance, have you ever wondered why enzymes, those remarkable biological catalysts, operate optimally at specific temperatures? Enzymes are sensitive to heat. As temperature rises, their activity typically increases, but too much heat can lead to denaturation, a loss of structure and function. Similarly, pH levels can dramatically affect enzymatic reactions. Each enzyme has a preferred pH range, and deviations can hinder its ability to perform. Concentration, whether it involves substrates or enzymes, also alters reaction dynamics. Picture a crowded marketplace. The more people there are, the busier it becomes. In a biochemical context, higher concentrations often lead to more frequent interactions, thereby accelerating reactions. But life does not merely thrive in comfortable environments. Many organisms exhibit extraordinary adaptations to extreme conditions. In the scorching heat of hot springs, for instance, thermophiles flourish, their proteins uniquely structured to withstand high temperatures. In contrast, extremophiles living in freezing climates have evolved antifreeze proteins that prevent ice crystal formation within their cells. The resilience of these organisms challenges our understanding of life's limits. As the microbiologist Carl Woese noted, life is much more robust than we ever imagined. This adaptability opens the door to exploring life in environments previously thought to be inhospitable, such as the icy moons of Jupiter or the sulfuric lakes of Venus. Yet, the delicate balance of biochemical processes is susceptible to disruption, particularly through pollutants. Have you ever considered how toxins affect the intricate web of life? Heavy metals, pesticides, and plastics seep into our environments, influencing biochemical pathways. For instance, mercury can inhibit enzyme functions, leading to detrimental health effects in both wildlife and humans. The impact of pollutants extends beyond individual organisms. Entire ecosystems can suffer when their biochemical foundations are compromised. As environmentalist Rachel Carson eloquently expressed, "In every outthrust headland, in every curving bay, in every grain of sand, there is the story of the earth." This story is increasingly marred by human actions that introduce harmful substances into our natural world. The specter of climate change looms ominously over the field of biochemistry, affecting not only the planet's temperature but also the very fabric of life. As temperatures rise, the delicate balance of ecosystems is disrupted. Species that once thrived in specific climates may find themselves struggling to survive. Have you imagined what happens to the intricate relationships between plants and pollinators as seasons shift? The timing of flowering plants and the emergence of pollinators is finely tuned, and any disruption can lead to mismatches that threaten survival. Moreover, ocean acidification, a direct consequence of increased carbon dioxide levels, alters the chemistry of marine environments. Organisms such as corals and shellfish rely on calcium carbonate to build their structures, and as the oceans become more acidic, these processes become more challenging. Picture a coral reef, once vibrant and teeming with life, now fading under the weight of changing conditions. The consequences extend to entire marine food chains, as those foundational organisms struggle to survive in their altered habitat. As we ponder the role of environmental factors in biochemistry, we recognize the intricate relationships that define life on Earth. Each element, from the sunlight that fuels photosynthesis to the pollutants that threaten our ecosystems, plays a critical role in shaping the biochemical landscape. The resilience of life, embodied in its ability to adapt and survive, inspires awe. Yet it also serves as a reminder of the responsibility we bear in nurturing the environments that sustain us. How will we choose to protect this delicate balance? The choices we make today will echo through generations to come. As we explore the vital process of photosynthesis, we find ourselves immersed in the remarkable ability of plants, algae, and certain bacteria to harness sunlight, converting it into chemical energy. This process is not merely a biological curiosity, it is the foundation of life on Earth. Imagine the sun's rays cascading down to illuminate vast fields, dense forests, and the shimmering surfaces of oceans, all serving as a catalyst for life. Through photosynthesis, these organisms capture solar energy and transform it into glucose, a sugar that fuels their growth and sustains countless forms of life that depend on them. At the heart of photosynthesis lies the chloroplast, a cellular powerhouse filled with chlorophyll, the pigment that gives plants their vibrant green hue. Within these organelles, a series of biochemical reactions unfold, beginning with the absorption of sunlight. As the light energy excites electrons, it initiates a chain of events that ultimately leads to the production of glucose and oxygen. The oxygen, a byproduct of this process, is released into the atmosphere, enriching the air we breathe. As Albert Einstein once mused, "Look deep into nature, and then you will understand everything better." Consider the intricate interplay of environmental factors that influence this process. Temperature, for instance, plays a crucial role in the efficiency of photosynthesis. Warmer temperatures can accelerate the rate of biochemical reactions, but there is a delicate balance. Excessive heat can lead to stress and even damage to the plant's cellular machinery. Have you ever felt the gentle warmth of the sun on your skin? Invigorating yet, when too intense, overwhelming. Plants experience a similar duality, as they thrive in optimal conditions but can falter under extreme heat. The pH of the environment also significantly impacts photosynthesis. Most plants prefer a slightly acidic to neutral soil pH, allowing for optimal nutrient availability. Imagine the subtle balance of elements in the soil, each contributing to the health of a plant. When the pH drifts too far from this ideal range, nutrient uptake may become impaired, leading to stunted growth and diminished health. Concentration gradients are yet another key factor. Nutrient concentration within the soil and water influences how effectively plants can absorb the minerals essential for their biochemical processes. Picture a vibrant garden flourishing because of rich, nutrient-dense soil, contrasted against barren land where nutrients are scarce. This discrepancy highlights the importance of environmental conditions in shaping the biochemical pathways that sustain life. As we delve deeper into the realm of biochemistry, we encounter organisms that thrive in extreme environments, known as extremophiles. These remarkable beings challenge our understanding of life's boundaries. From the scorching heat of hydrothermal vents to the freezing temperatures of polar ice, extremophiles adapt in extraordinary ways. Take, for example, the thermophiles, which flourish in temperatures well above the boiling point of water. Their enzymes, adapted to function at such extremes, have become invaluable in scientific applications. The discovery of Taq polymerase, an enzyme derived from a thermophile, revolutionized molecular biology, enabling the polymerase chain reaction, a technique that amplifies DNA. As microbiologist Thomas Brock, who discovered this enzyme, stated, "We learned that life can exist under conditions that we would consider hostile." In contrast, psychrophiles thrive in icy environments, their biochemical processes uniquely adapted to low temperatures. These organisms produce antifreeze proteins that prevent ice crystal formation, allowing them to survive and flourish where most life would perish. Imagine the resilience of life as it bends and adapts to the unyielding forces of nature, showcasing an extraordinary spectrum of biochemical ingenuity. Yet as we marvel at these adaptations, we must also confront the harsh reality of environmental pollutants and their impact on biochemical processes. Human activity has introduced a plethora of chemicals into our ecosystems, altering the delicate balance that sustains life. Heavy metals, pesticides, and plastics infiltrate our soils and waterways, posing significant threats to organisms. Consider the plight of aquatic life as they navigate through waters tainted by pollutants. The biochemical pathways that enable fish and amphibians to thrive can be disrupted by toxins, leading to impaired growth, reproductive challenges, and even mortality. The story of the mute swan is telling. Once abundant in the waters of Europe, their populations dwindled due to lead poisoning from fishing gear. As conservationist and biologist Dr. Sylvia A. Earle poignantly remarked, "No water, no life. No blue, no green." In our pursuit of progress, we often overlook the consequences of our actions on the intricate web of life. The introduction of pollutants into ecosystems is not merely a matter of environmental concern. It directly impacts the biochemical processes that drive life itself. As we gaze toward the horizon, the specter of climate change looms large, casting shadows over the delicate balance of biochemistry and life. Rising temperatures, altered precipitation patterns, and shifting ecosystems all result from our changing climate. These changes reverberate through the biochemical pathways that underpin life, leading to profound consequences for organisms across the globe. Imagine a coral reef, once a bustling hub of biodiversity, now struggling to survive as ocean temperatures rise. Coral bleaching, a phenomenon driven by increased water temperatures, disrupts the symbiotic relationships between corals and the algae living within them. These algae provide essential nutrients through photosynthesis, and when corals become stressed, they expel these vital partners. The vibrant colors fade, and the once thriving ecosystem is left vulnerable and bare. The repercussions extend beyond the reef itself. As the foundation of marine food webs, corals support a multitude of species, from fish to crustaceans. When these foundational organisms falter, the entire ecosystem suffers. The words of renowned marine biologist Dr. Carl Safina resonate. The ocean is a source of life, and the health of the planet depends on the health of the ocean. This intricate interplay of environmental factors and biochemistry highlights the profound connections between life and its surroundings. Each organism, from the smallest microbe to the largest mammal, is intricately woven into the fabric of its environment. The delicate balance that sustains life is a reminder of the responsibility we carry as stewards of the earth. As we ponder the future, we must ask ourselves how we can foster resilience in the face of these challenges. How can we protect the environments that support the biochemical processes of life? The answers lie not only in scientific inquiry, but also in our collective actions and choices. Through sustainable practices, conservation efforts, and a deeper understanding of the interdependence of life, we can begin to restore the balance that sustains us all. In this symphony of life, the notes of resilience, adaptation, and responsibility echo through time. The intricate relationships that define our world remind us of the beauty and fragility of existence. What will the next chapter in this story hold? The answer lies in our hands, as we navigate the delicate balance of nature and nurture the environments that cradle life itself. As we reflect on the urgent need to protect and nurture the ecosystems that support biochemical processes, we find ourselves at a pivotal juncture. The challenges posed by environmental changes not only compel us to act, but also inspire innovation in the life sciences. Advancements in genetic engineering and synthetic biology promise new pathways to restore balance and enhance resilience within our ecosystems. In the following chapter, we will explore these exciting developments and their implications for medicine, health, and the ethical landscape of biochemistry. As we delve into the future directions of this vibrant field, we invite you to consider how these innovations can shape a more sustainable and harmonious relationship between humanity and the natural world. Chapter 10. Future Directions in Biochemistry and Life Sciences. The world of biochemistry is a vast, intricate web of pathways, all interconnected in ways that can astonish the mind. Imagine a bustling city where every street leads to another, where each neighborhood has its own unique character yet contributes to the whole. This is akin to the biochemical pathways in living organisms. Each pathway, whether it deals with metabolism, signal transduction, or cellular repair, interacts with others to create a harmonious balance essential for life. The interconnectedness of these pathways reflects the complexity of life itself. Biochemical reactions do not occur in isolation, but are part of a grander system. For instance, the way in which glucose is metabolized for energy also affects the synthesis of amino acids and nucleotides. As the renowned biochemist Albert Szent-Györgyi once said, "Discovery is seeing what everybody has seen and thinking what nobody has thought." This idea resonates deeply in the study of biochemical pathways, where the relationships between different reactions and molecules reveal insights into how life functions on a cellular level. Advances in genetic engineering and synthetic biology have propelled our understanding and manipulation of these pathways to unprecedented heights. Genetic engineering allows scientists to alter the genetic makeup of organisms, creating new traits and functions that nature alone could not achieve. Imagine a world where crops are engineered to be more resilient to climate change, or where bacteria are designed to produce insulin more efficiently. The possibilities are vast and inspiring. Synthetic biology, on the other hand, seeks to construct new biological parts and systems, or to redesign existing ones for useful purposes. This field is akin to a biological Lego set, allowing scientists to assemble life's building blocks in novel configurations. Jennifer Doudna, a pioneer in CRISPR technology, has expressed, "We have the potential to make changes that could improve health and address global challenges." As we delve deeper into the role of biochemistry in medicine and health innovations, the impact becomes increasingly evident. This field has been at the forefront of numerous breakthroughs, from the development of targeted therapies for cancer to the creation of personalized medicine tailored to an individual's genetic profile. Picture this: a world where treatments are not one-size-fits-all, but are specifically designed for your unique genetic structure. This is not a distant dream, but an emerging reality. The precision of biochemistry allows us to understand the mechanisms of disease at a molecular level, leading to more effective and less invasive treatments. Consider the words of Francis Collins, director of the National Institutes of Health, who stated, "We have a remarkable opportunity to translate our understanding of genetics into effective treatment for diseases." This translation from basic research to practical health solutions embodies the core of biochemistry's role in modern medicine. As we look towards emerging fields such as metabolomics and systems biology, we find new dimensions of understanding unfolding. Metabolomics studies the unique chemical fingerprints that cellular processes leave behind, encompassing a wide array of metabolites that provide insights into health and disease states. Imagine a detailed map of the body's metabolic landscape, where the fluctuations in metabolites can indicate the onset of disease long before symptoms appear. Systems biology complements this by integrating data from various biological levels, from genes to proteins to metabolites. This holistic approach allows scientists to see the bigger picture, understanding how changes in one part of the system can ripple through the entire organism. As systems biologist Leroy Hood has articulated, the future of medicine is in understanding complex systems. This understanding opens the door to preventative medicine, where interventions can be made before conditions become serious. With progress comes the necessity for ethical considerations in biochemistry research and applications. As we innovate and explore the frontiers of genetic engineering, synthetic biology, and personalized medicine, we must also grapple with the moral implications of our work. Questions arise. How far should we go in altering the genetic fabric of life? What are the ramifications of creating synthetic organisms? The debate surrounding these issues is vibrant and essential. Ethical frameworks must guide biochemists and researchers through these uncharted waters. The words of geneticist and ethicist Paul Berg resonate here. Our ability to change genes and alter life forms is an awesome responsibility. It is crucial to establish dialogue among scientists, ethicists, policymakers, and the public to ensure that advancements serve humanity as a whole. Transparency, respect for life, and consideration for potential consequences are paramount in the pursuit of knowledge and innovation. As we contemplate the future directions in biochemistry and life sciences, we can sense the excitement and possibilities that lie ahead. The interconnectedness of biochemical pathways, the leaps in genetic engineering, the transformative role of biochemistry in health, the emergence of metabolomics in systems biology, and the ethical considerations surrounding these advancements weave together a complex narrative. This narrative is not just about scientific progress, it is about the very fabric of life itself. Each discovery, each technological advancement, brings us closer to understanding how to nurture health, combat diseases, and respect the intricate balance of ecosystems. Imagine a future where biochemistry continues to illuminate the path of discovery, where the beauty of life is appreciated in all its complexity, and where the ethical considerations of our actions shape a better world for generations to come. The journey of understanding has only just begun, and the chapters yet to be written hold the promise of profound change. The interconnectedness of biochemical pathways and organisms serves as a reminder of the intricate web of life that surrounds us. Consider the vast network of interactions that occurs within a single cell, where various pathways converge and diverge, creating a symphony of biochemical activity. Each pathway plays a specific role, contributing to the overall functioning of the organism. Imagine, for a moment, that you are a part of this network, where every action influences another, creating a balance essential for survival. One of the most intriguing aspects of this interconnectedness is how biochemical pathways can adapt to environmental changes. For instance, when faced with stress, such as oxidative damage or nutrient deprivation, cells can reroute their biochemical pathways to generate the necessary energy or repair mechanisms. This adaptability is not merely a survival tactic, it is a testament to the evolutionary sophistication of life itself. As biochemist and Nobel laureate Frances Arnold noted, nature is a remarkable engineer. It has solved many problems we are only beginning to understand. As we delve deeper into the future of biochemistry, the advancements in genetic engineering and synthetic biology stand at the forefront. These fields are revolutionizing our understanding of biological systems and opening doors that were once thought to be closed. Techniques such as CRISPR-Cas9 have empowered researchers to edit genomes with unprecedented precision, allowing for the correction of genetic defects and the enhancement of beneficial traits. Picture a world where diseases caused by single gene mutations can be corrected at their source, offering hope to millions. Yet, with such power comes the question of responsibility. What guidelines should govern the use of these technologies? The potential to create genetically modified organisms or even synthetic life forms raises profound ethical dilemmas. As biochemist Jennifer Doudna, one of the pioneers of CRISPR technology, poignantly states, we need to be careful about how we apply these tools. We have to think about the consequences. In parallel, the role of biochemistry in medicine and health innovations is expanding at an extraordinary pace. The development of targeted therapies, personalized medicine, and innovative drug delivery systems exemplifies how biochemistry is reshaping patient care. Imagine a future where treatments are tailored to the unique biochemical makeup of each individual, maximizing efficacy while minimizing side effects. The burgeoning field of metabolomics adds another layer of complexity to our understanding of health and disease. By analyzing the unique metabolic profiles of individuals, scientists can gain insights into their health status, uncovering biomarkers that indicate disease onset or progression. This approach allows for early intervention and more effective treatment strategies. The words of renowned metabolomics researcher Gary Patty resonate here. Metabolomics offers a window into cellular function and can help us understand not just disease, but health itself. Systems biology, too, is emerging as a powerful paradigm that complements our grasp of biochemistry. It integrates data from genomics, proteomics, metabolomics, and other disciplines to create a holistic view of biological systems. By understanding how these components interact, researchers can elucidate the underlying mechanisms of complex diseases, such as cancer or neurodegenerative disorders. Imagine the possibilities that arise when we can model biological systems in a way that mirrors their true complexity, allowing for a more comprehensive approach to research and treatment. As we explore these promising advancements, it is essential to approach them with a sense of caution and ethical responsibility. The convergence of biochemistry and cutting-edge technology invites us to consider the implications of our actions. Should we engineer organisms for agricultural enhancement, or do we risk disrupting ecosystems that have evolved over millennia? What happens when we create synthetic life? Are we crossing a threshold that should remain uncrossed? These questions linger, urging us to engage in thoughtful discourse. The ethical considerations surrounding biochemistry research and applications cannot be overstated. As we venture into uncharted territories, the need for robust ethical frameworks becomes increasingly critical. Dialogue among scientists, ethicists, and the public is essential to navigate the complexities of these advancements. The words of philosopher and bioethicist Peter Singer remind us, the challenge is to balance the benefits of scientific progress with the moral implications of our choices. In this rapidly evolving landscape, we must ensure that our pursuit of knowledge aligns with the greater good. Transparency in research, respect for diverse perspectives, and an unwavering commitment to ethical standards will guide us through the maze of possibilities. Imagine a future where biochemistry serves as a force for good, addressing global challenges such as food security, climate change, and health disparities. As we contemplate the future directions in biochemistry and life sciences, the excitement is palpable. The interconnectedness of biochemical pathways, the advancements in genetic engineering, the transformative role of biochemistry in health, the emergence of metabolomics and systems biology, and the ethical considerations surrounding these innovations all weave together a narrative rich with potential. Each discovery invites us to reflect on our place within this intricate web of life, urging us to embrace the responsibility that comes with knowledge. The journey of discovery is ongoing, and as we continue to unlock the secrets of life at the molecular level, we find ourselves on the brink of profound change. The chapters yet to be written hold the promise of a deeper understanding of ourselves and the world around us. With each advancement, we inch closer to a future where biochemistry not only enhances our lives, but also respects the delicate balance that sustains all living things. As we stand at the crossroads of biochemistry and life sciences, the horizon gleams with promise and potential, inviting us to tread thoughtfully into the unknown. The intricate dance of molecular interactions, paired with our ethical responsibilities, shapes not only our understanding of life, but also the legacy we leave behind. By fostering a culture of transparency and collaboration, we can harness these advancements to address pressing global challenges while honoring the delicate ecosystems that sustain us. In embracing this duality of innovation and integrity, we prepare to embark on a transformative journey that could redefine the very essence of our existence. With each stride forward, we lay the groundwork for a future where biochemistry truly serves as a beacon of hope and healing, illuminating the path for generations to come.